Hello, everyone, and welcome to today's live broadcast, New Tools for Understanding Allosteric Signaling in G-Protein-Coupled Receptors, presented by Matthew Eddy, Ph.D., a postdoctoral research fellow at the Scripps Research Institute. I'm Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen. Or you can use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the accreditation button located in the promotional board at the bottom center of your screen and follow the process to obtain your credits. Please join me in welcoming Dr. Eddy. I will now turn the presentation over to him.
Thank you very much. So today I'm excited to talk to you about my research and specifically about mechanisms of allosteric signaling in human GPCRs. So to provide an overview of what I'm going to present today, first, I'm going to provide an introduction to human GPCRs and a brief overview of their structure, function, and physiological relevance. And then I want to tell you a little bit about their relevance for drug discovery. The second and third topics belong under the same umbrella, and that is integrative structural biology techniques to study GPCR structure and function. In the first of these two, we'll be talking about allosteric communication in human GPCRs studied by X-ray crystallography, and specifically the role of an allosteric center as studied by X-ray crystallography. And in the third and final section, we're going to be talking about allosteric communication in human GPCRs as studied by NMR spectroscopy. And specifically, I will tell you about how solution nuclear magnetic resonance, or NMR, reveals allosteric pathways in human GPCRs. So first, I'd like to provide an overview of GPCR structure and function, or G-protein-coupled receptors. On this slide, you see a cartoon of the cell surface in which the lipid bilayer is represented by these gray rectangles. And the GPCR is shown here in a red cartoon passing through that lipid bilayer on the cell surface. It has a 7-transmembrane or 7-TM architecture. Inside of the cell, below the intracellular surface, you see also a trimeric G-protein complex, which I will describe its interactions with the GPCR on the following slide. Now, the basic scheme of action for human GPCRs at the cell surface proceeds as follows. So you have a small molecule, in this case adrenaline. It's recognized by the GPCR and it binds to it, causing a conformational change to happen on the intracellular surface. This change enables complex formation with the trimeric G protein that facilitates exchange between GTP and GDP, shown here. This exchange then breaks apart this trimeric G protein complex into 2 subunits that each activate signaling cascades. This ultimately results in a physiological consequence. Now, in humans, we have 826 different GPCRs, all of which play critical roles in human health and diseases. And here are just some examples of the areas where human GPCRs impact physiology and health. So our vision is mediated by rhodopsin, which is a human GPCR. Cardiovascular health is mediated by a number of different GPCRs, including adrenergic receptors, which are responsible for the fight or flight mechanism, adenosine receptors, and so on. Allergies are mediated by histamine receptors. Metabolic health and illnesses are also mediated by GPCRs. Immune system function, GPCRs play a critical role in this, as well as critical roles in how diseases hijack and take advantage of our immune systems. And GPCRs play critical roles in the health of the central nervous system, including feeling of pain. So GPCRs are really how we interact with our environment around us. They're responsible for our sight, our smell, that's olfactory receptors, taste receptors. Even the way we think and feel and consciousness of self may be mediated by human GPCRs. Human GPCRs are also major drug targets. And on this slide, you see some examples of some of the most widely used and perhaps abused drugs that target GPCRs. So we have caffeine present in coffee, theobromine, which is present in chocolate, and theophylline present in tea. Now, in terms of drug discovery, GPCRs have historically been a very popular target for treating illnesses and developing new drugs to treat diseases. So currently about 35 to 40% of FDA-approved drugs target human G-protein coupled receptors. There have been 116 GPCR-targeted drugs approved in the last 20 years, and there appears to be no decline in the success of human GPCR drug discovery projecting forward. Out of those 826 unique human GPCRs, so far only 43 have been successfully targeted for drug discovery. Thus, there are remaining many potential GPCRs that could be great targets for treating new diseases. On this slide, you see just a few examples of a number of commercial FDA-approved drugs. Probably many of you are taking one or more of these currently. So GPCRs are major drug targets and will continue to be so. And since we're talking about drugs, it's useful to talk about what they actually do in terms of affecting the GPCR function and structure. And we typically classify GPCR drugs or ligands according to their function or efficacy. And so in here, all GPCRs have what we call basal level of signaling. And this is represented on the left side of the slide. And you see a kind of a straight line that goes across from the left to the right of the chart. Now, some drugs are known as neutral antagonists, and so they will bind and be recognized by GPCRs, but they don't change the level of basal signaling. Some drugs bind to GPCRs and activate the GPCR, or they increase that level of signaling. We call these agonists, and you can either have a full agonist, which leads to full or maximal activation, or a partial agonist, which is a drug that binds to the receptor, causing sub-maximal activation. And finally, the last class of orthosteric binding drugs are known as inverse agonists, in which they bind to the GPCR and they actually lower the level of signaling below the basal level of activity. So now what is the consequence of these drugs binding to the receptor in terms of their structure? Well, we solved a number of different GPCR structures with X-ray crystallography for different kinds of drugs of different efficacies. And here are just two examples. Now, here we have on the right side of the slide, at the top is a side view of a superposition of two crystal structures of the adenosine A2A receptor. This is in complex with an antagonist, and then also in complex with an agonist. And on the bottom, you see a superposition of the same two structures from an intracellular view, here indicating the change in the orientation of the helices differences in the efficacies of the ligand. This change in the orientation of these helices and this motion is what leads to and enables complex formation with the intracellular G protein partner. Now, one of the questions that really intrigued me is, what is the mechanism of allosteric communication between the drug binding pocket and the intracellular surface? So on this slide, we see an example of a GPCR G protein complex, and the drug is shown in a space-filling representation toward the top of the blue GPCR near the extracellular surface. So the distance between that binding site and the site on the intracellular surface where the G protein forms a complex with the GPCR is about 30 angstroms away, which is quite a far distance on a microscopic scale. So it's of great interest to me how this signal is propagated through the receptor to cause these conformational changes on the intracellular surface of the receptor, ultimately leading to complex formation with the partner protein inside of the cell. And that's what motivated my research. Now I want to get into the heart of the talk and present new research attempting to answer this question. And first, I'm going to talk about allosteric communication in human GPCRs as studied by X-ray crystallography, and in particular, the role of an allosteric center as studied by X-ray crystallography. And for this work, we focused on one particular GPCR. It's called human adenosine A2A receptor, or A2A-AR. Now, the adenosine A2A receptor is one of the main targets for caffeine, theobromine, and theophylline, the drugs that I had mentioned on an earlier slide. In addition to that, it's also responsible for mediating myocardial blood flow, dopamine release, and immune system regulation. It's a validated target for treating Parkinson's disease, and it's currently being explored by different companies as a cancer co-immunotherapy target as well. So it's really important for human health and disease. On the right, you see an example of a crystal structure of the human adenosine A2A receptor, shown in green, PDB3EML here. You'll notice that there's the 7TM or transmembrane architecture with the ligand bound to the top in the orthosteric binding pocket. In addition to that, toward the bottom of the structure, on the left-hand side, you'll notice there's an additional protein domain. This domain is not present naturally, and it's added in order to crystallize the receptor to obtain its structure. I'll talk about a little bit about the consequences of this in subsequent slides. Now, to get at this question of how signals are propagated through the receptor, there's a second clue that we wanted to focus on. And the role of a particular residue in an allosteric center. This is aspartic acid 52. On the left is a crystal structure, a high-resolution 1.8 angstrom crystal structure of the human A2A receptor. And you'll see all these red spheres. The red spheres are crystallographic water. Now, if we expand the central region in the structure in the center of this slide, you'll see there are several residues that form coordinative bonds with these waters, as well as with the blue sphere at the center, which is a sodium ion. Aspartic acid 52 is at the bottom of this cluster, and this residue is highly conserved among all class A GPCRs, which is the rhodopsin class of GPCRs, and which is a majority of GPCRs. Now, something like 85% or more different class A GPCRs have this aspartic acid 52, this charged residue. If they don't, they typically have another charged residue, such as glutamic acid, in its place. So it's highly conserved. Now, if we replace this aspartic acid 52 with the neutral asparagine, this leads to a variant protein with some interesting properties. So on the left is a cartoon showing that for the wild type or native A2AAR, when an agonist or ligand binds to the receptor, we get G protein signaling as a consequence of that. For this particular variant, D52N, it doesn't perturb ligand binding, so ligands bind like they would for the native protein, but there's absolutely no G protein signaling. So you can bind an agonist, and G protein signaling is completely killed for this variant. So this phenomena is also widely observed and been reported for other GPCRs with this particular variant. So it seems critical that this is involved with signaling pathways in not just the A2A receptor, but other class A GPCRs as well. Now, the cartoon on the previous slide is also supported on this slide, which is showing the actual data. So on the left, you can see radioligand binding data. So we take a drug, it's labeled with tritium, and observe its binding activity or affinity for a particular target. In this case, we looked at an agonist, and you can see that the data we measure for the wild type protein is almost identical for this special D52N variant protein. Whereas on the right, we have a G protein signaling assay. And so you can see in gray, the native protein, when we add an agonist, we get a response, an upward response on this agonist efficacy curve, indicating G protein signaling. Whereas for the D52N variant, we get no signaling whatsoever, even with very high concentration of agonist. Together with my co-author, Kate White, we solved 2.5 angstrom crystal structure of the variant protein AZAAR D52N in complex with an agonist, which was recently published in the journal Structure. And this complex is presented here. So you can see it has the 7TM architecture like we would expect, and the ligand is shown bound to the top of the receptor in a vertical binding pose. Now what's interesting is if we compare this crystal structure with the native protein in complex with the same agonist and with a ligand of different pharmacological efficacy. On the left, we see a side group of the variant protein D52N in complex with this agonist UK432.097. And that's superimposed over the structure of the native protein in complex with an antagonist, that's in yellow. And you can see that there are subtle differences between the two. On the right, we have a superposition of the variant protein in complex with the agonist, again, superimposed with the native protein in complex with the same agonist, and the structures are essentially identical from this view. So that comparison is a little bit better illustrated in this RMSD plot of the superposition of the two proteins. And so you can see at the top, in the top panel, when we're comparing the differences between the structures, there's essentially no difference, except for a region that is local to the site of the mutation, and that's expanded in the lower box there. So that's this residue serine 7.46 that you see, valine 7.47, and so on. So there we see the largest structural deviations between the variant protein D52N and the native protein. It turns out that that region that we see the largest difference is close to a conserved activation motif that is conserved among most class A GPCRs. It's called the NPXXY activation motif. And in this slide here, we're looking at several different conserved activation motifs, which I won't get into in detail. The important point is the one that's highlighted in cyan on the lower right. It's the NPXXY motif, which is where we actually see structural deviations. Now, in the bottom figure, this is a cartoon representing hydrogen bonds between 5 of the 7 helices near this activation microswitch region. And this cartoon is comparing changes in hydrogen bonding. So on the left, we have hydrogen bonding for the antagonist complex, in which you see we have hydrogen bonds between helices 1 and 7 and 2 and 7. And you can contrast that to the middle diagram, where we see changes in the hydrogen bonding between helices 7 and 6 and 2 and 3. And hydrogen bonding observed for the agonist complex with the variant protein D52N, in which we see a kind of high between the two. So some indication of hydrogen bonding observed from the antagonist complex, and a little bit of hydrogen bonding observed from the agonist complex, so between helices 2 and 3. So these changes in hydrogen bonding are what we propose contribute to differences in signaling between the two proteins. However, what was interesting also was the fact that the structures were so highly similar. Now, on this slide here, we then have the same superpositions of the variant D52N agonist protein complex with the antagonist complex shown on the left, and a superposition with the native protein agonist complex shown on the right. And the bottom two figures are, again, intracellular views, so inside the cell looking up at superpositions of the two proteins. On the left, we see changes in the relative orientations of the helices. On the right, we see essentially no change whatsoever. Now, this is interesting because at the intracellular surface, there's no signaling observed for the D52N variant protein, whereas the native protein does signal. So from this view, the two structures are the same, and in that respect don't fully explain the differences that we see with the data. So that motivated us to think about the role of conformational dynamics and its relationship to GPCR function. And to introduce this topic, I really like this quote from Richard Feynman, who says, "Everything that living things do can be understood in terms of the jiggling and wiggling of atoms." And here we have a comparison of some of the strengths between X-ray crystallography and NMR, whereas X-ray crystallography can give us very high resolution information. That information is of a single conformational state. Whereas NMR spectroscopy, with NMR spectroscopy, we can observe multiple conformations that are simultaneously populated and in equilibrium. So with that, I would like to come to the third point of the presentation, being investigating allosteric communication in human GPCRs by NMR spectroscopy. And in particular, I'm going to talk about how solution NMR spectroscopy reveals allosteric pathways in the human adenosine A2A receptor. Now, when I had started my postdoctoral studies with Ray Stevens and Kurt Wüthrich, this slide represents some examples of state-of-the-art, at the time, studies of human GPCRs. So on the left, for example, there was studies using fluorine NMR spectroscopy to study the beta-2 adrenergic receptor. This is one example from the Wüthrich lab. Using fluorine NMR, simple one-dimensional NMR experiments to study bias signaling in the human beta-2 adrenergic receptor. On the right, there was a technique using reductive methylation that was first published in its relevancy to studying human GPCRs by the Kabilka group. And this is an example of the kind of data that was obtained with this method. And so lysines could be methylated with a carbon-13 CH3 group and thereby studied with NMR spectroscopy. So what these studies had in common is that they typically introduce isotopic labels, which are necessary for NMR spectroscopy, as a kind of post-translational modification. And the reasons for that are really that, first, production and purification of human GPCRs is challenging, regardless of whatever technique you use to study its structure, function, and dynamics. But second, that incorporation of the stable isotopes required for NMR spectroscopy is also challenging and adds further difficulties to the experiments. So my own research and interest is in trying to expand the number of NMR probes that we use to study GPCRs in order to provide a more comprehensive view of GPCR structure and function. This is illustrated by this cartoon on this slide. So we had seen a number of different GPCR studies where there were a few probes introduced by chemical modification or a kind of post-translational modification. This has well-established biochemical methods. However, it provides less comprehensive view of signaling. And what I'm interested in is providing a more comprehensive view by introducing NMR probes through biosynthetic incorporation. So this is more challenging for a number of reasons, but we typically take the approach of uniform incorporation of stable nitrogen-15, carbon-13, and deuterium isotopes directly into the GPCR expression media. And for this, we're expressing our protein in yeast, or Pichia pastoris. Most of the crystallography and other structural studies done on human GPCRs have expressed the receptor in either insect or mammalian cell systems. Because of that, we wanted to carefully characterize the material that we expressed to make sure that its function and fold and properties were identical to the same protein expressed in insect or mammalian cell systems. On the left, you see some representative radioligand binding assays indicating that we observe native-like ligand binding for the receptor expressed in yeast membranes as compared to mammalian cell expression systems. On the right, in the bottom right, you see an example analytical size exclusion chromatography trace of isotopically labeled human A2A AR expressed in yeast just prior to NMR studies. So we see essentially a single, well-resolved component indicating a homogeneous and monodispersed sample. On the top right, you see these are so-called fluorescence thermal shift assays, which we use to indirectly monitor ligand binding to GPCRs. So these S-shaped curves you see shift to the right, indicating ligand binding to the receptor, increasing its melting temperature, indirectly also validating the ligand binding assays. Now, on this slide, unfortunately, this slide looks a little bit distorted. The main point is that we directly monitored radioligand binding of the samples that we prepared for NMR studies in the conditions used for NMR studies, and that being A2A AR reconstituted into detergent micelles. So we used scintillation proximity assays and directly measured the ligand binding of several different ligands to the receptor in the same conditions used for NMR studies, and we could validate that the protein retained its native-like function in the same conditions we used for all of our experiments. Now we further validated our system by expressing a so-called GPCR fusion construct in Pichia pastoris, the same strain that we were using for production for NMR studies, and we crystallized that protein. So some of the crystals of that are shown on the left, and on the right, you see a superposition of the structure of the A2AAR fusion protein in complex with an antagonist, and it's superimposed with an earlier structure of the same protein as expressed in insect cells, and overall the two are essentially identical. Now, importantly for NMR studies, we did not use a fusion protein. There were no stabilizing point mutations introduced in the receptor. The protein is reconstituted into mixed micelles containing cholesterol hemisuccinate that retain native-like binding properties as demonstrated by radioligand binding assays. Now, after much optimization of a number of different conditions used for expressing the protein as well as for obtaining NMR data, we were able to obtain very high resolution solution NMR data for the adenosine A2A receptor. Shown on this slide is a representative NMR spectrum. This is a TROSY spectrum as measured at 800 megahertz, in which we see over 270 individually resolved signals. And this was recorded with only a few milligrams of protein in less than a day at 800 megahertz without a cryoprobe. Now, as a point of reference, I present the spectrum from the previous slide and compare that to some published NMR data from the Gerhard Wagner laboratory, in which they measured TROSY NMR spectra of a protein expressed in E. coli. This is bacteria rhodopsin, which is also a 7-TM protein, but not a GPCR. And you can see at first glance the quality of the two spectra are comparable, indicating that we have, again, very high quality, high resolution NMR data that we can use to learn something new about the protein. So why did we use Pichia pastores and go to such great effort to validate the expression system? And the reason is simple. We can incorporate deuterium oxide into the expression media for yeast. So yeast tolerate high levels of D2O, and the D2O is necessary to obtain the high resolution spectra that you see. So on this slide, we compare the effects of a uniformly nitrogen-15 labeled sample with and without deuteration. In the top spectrum, you can see this is the native A2AAR without deuteration. And in the bottom spectrum, the only difference here is that this spectrum was recorded for a protein with deuteration. So this this was the motivation for using the yeast expression system. So now that we have this nice NMR data of the adenosine A2A receptor, we want to be able to identify and assign some of those well-resolved signals. And each one of those signals then is arising from an individual proton-nitrogen pair. And in order to assign and identify them, we took an approach where we first we looked at the NMR spectrum, and in particular focused on two characteristic regions. So one is the tryptophan indole region, which is located in the lower left, and the other is the glycine backbone region, which tends to appear toward the top of the NMR spectrum. In order to assign these signals, we used amino acid replacement, and thereby replacing one by one a glycine with an alanine, or a tryptophan with a phenylalanine or a tyrosine. And the summary of all of those experiments are shown on the right-hand side of this slide. So you can see, for example, for tryptophan 29, which is located toward the top of the panel of data, we replaced that with a tyrosine, and we were able to then unambiguously assign the signal from tryptophan 29 that way, as indicated by the star and the traces through the NMR data in each panel. By doing so, we were able to assign all 6 tryptophans located on the extracellular and intracellular surfaces, as well as the membrane spanning regions. And we were also able to further find 8 glycines distributed on the extracellular surface and the intracellular surface, including at a key location on the intracellular end of Helix 6. As you remember from the title slide, there was a picture of the receptor with lanterns hung in key positions. And this was a motivation for that picture, suggesting that each one of these lanterns is an NMR probe lighting up local structural regions of the receptor, and altogether providing a comprehensive view of the protein. Now that we have assigned and identified these signals, we can start to ask questions about how do the NMR data or signals respond to changes in drug efficacy. So on this slide, you see on the left, we have panels from two-dimensional TROSY NMR experiments in which each panel we have focused on the indole tryptophan region of the spectrum for 6 different complexes. So 3 complexes with antagonists, shown on the left, and 3 complexes with agonists, shown on the right. And these boxes, dashed and dotted boxes, are important as well, and we'll get to them on the next slide. So if you just compare, for example, at the top of this panel of 6, the data with the antagonist and agonist complexes, we see changes in the peak shapes as well as their positions or chemical shifts, indicating responses to variable drug efficacy or changes in drug efficacy. And we can better visualize some of those responses by looking at projections. So on this slide, we have projections through the boxes that I showed on the previous slide. And we have the signals assigned and annotated in each of these boxes. So at the top, you can see there's data projections for the tryptophan signals for the complex with the antagonist CM241385. Now, what is interesting to compare are the shape and intensities of signals between the tryptophans on the intracellular surface versus the extracellular surface. So you can see the signals for tryptophan 29, there are actually 2 of them, and they're much broader compared to the signal for tryptophan 143, which is located on the extracellular surface, which shows a single signal that is relatively narrower than what's observed for tryptophan 29. You'll also notice for the complexes with antagonists, so there are 2 examples in the bottom of this panel, that's NECA and UK432097. So tryptophan 32, for those complexes, actually we observe 2 components in the signal for tryptophan 32, and overall the signal is broader than that's what's observed for tryptophans on the extracellular surface. So this broadening or signal multiplicity is due to the presence of multiple simultaneously populated conformers that are local and in exchange. We can also now look at the glycines which we've assigned and see how they respond to variable drug efficacy or changes in drug efficacy. And this slide just shows some examples of that kind of comparison we can make. So for example, at the top, you'll see data for glycine 118, which is located on the intracellular surface at the end of Helix 4. And we see large changes there in chemical shift or position of the NMR signal that directly depends on the efficacy of the bound drug. We also see changes for glycine 142, which is located on the intracellular surface as well as changes for glycine 218 and 114. And I will get further into detail for those in the subsequent slides as it relates to function. What was one of the more interesting observations, again, was for glycine 118, which is located at the intracellular surface of helix 4. And we saw large changes in chemical shifts for the signals for glycine 118, indicating differences in the backbone conformation locally near glycine 118 that depends in a ligand efficacy dependent manner. So this was interesting because if you look at the crystal structures of the adenosine A2A receptor and focus on the region that's local to glycine 118, there are essentially no differences between those structures, whereas the NMR data indicate that there are larger differences that are observed for the solution experiments, indicating that NMR may provide some additional insight into changes in structure that are missed with crystallography. Now, perhaps one of the most interesting observations was made with tryptophan 246, which is known in the GPCR literature as a toggle switch tryptophan, going back to studies of rhodopsin. It turns out that tryptophan 246 is a highly sensitive probe of drug efficacy in the human adenosine A2A receptor. On the left, you see two-dimensional TROSY NMR spectra, focusing on the region where we identified tryptophan 246. In the top spectrum, for the complex with the antagonist CM241385, you see the location of tryptophan 246 is indicated by the line toward the right side of the window. In the bottom spectrum, we have a complex with the agonist NECA. And in this complex, we've assigned the signal for tryptophan 246, which is all the way over on the left side, lower left side of the window, roughly about 1 ppm difference in chemical shifts between the two complexes. This is a very large efficacy-dependent chemical shift that's observed for this toggle switch tryptophan. And what is the rationale for this large chemical shift? Well, when you look at the crystal structures of the adenosine A2A receptor, there's a nearby phenylalanine ring, and there's a well-known phenomenon in NMR spectroscopy of so-called ring current shifts. So if you have an aromatic group that is nearby an NMR probe, the relative position and orientation of that aromatic group can have a significant effect on the chemical shift of the observed NMR probe. And this goes back quite a long ways in NMR spectroscopy. As illustrated here on the right in this cartoon, the changes of the orientation and position of the ring relative to the NMR probe can have pronounced effects on the chemical shift of that probe. So we can also see that we observe similar changes when we compare other complexes. So again, the top two panels are between the antagonist ZM and agonist NECA. The bottom, again, is for the antagonist XAC, which has a different scaffold, and agonist CGS21680. So structurally, the origins of this large change that we see are due to nearby phenylalanine ring 242 and its position relative to the toggle switch tryptophan 246. What's really exciting is that this phenylalanine 242 is part of a conserved activation motif called the PIF activation motif, consisting of proline 189, isoleucine 92, and phenylalanine 242. So the relative changes in the conformation of activation motif and changes of the position or chemical shift of the indole signal for tryptophan 246. Now, this PIF activation motif is highly conserved. On the right is a figure from a previous paper from the Stevens lab, first indicating the importance of this activation motif and showing how the local conformation of these residues change between structures of with ligands of different efficacies. And we propose that this observation may be useful to study a number of different human GPCRs. For example, recently there were solved crystal structures of the human cannabinoid receptor in complex with drugs of different efficacies. So shown here are superpositions of a CB1 in complex with an agonist and antagonist, and a comparable residue, so-called toggle switch tryptophan, is shown here on an expansion of this, where it undergoes a large reorientation between the two complexes, which we would anticipate we would also see a large change in that chemical shift of that toggle switch tryptophan. So this observation could likely be extended to other class A GPCRs as well. So this brings me back to the central question of signal transduction in GPCRs, and in particular, what is special about this aspartic acid 52 and why this amino acid replacement D52N permits ligand binding and then kills G protein signaling. And this slide is just a reminder of that behavior that we see for the variant protein. Now, when we look at this toggle switch tryptophan and compare the data between the native protein and the variant D52N, we see some interesting differences. So on the left, the blue spectra that you see are for the native protein, and we see the location of the toggle switch tryptophan as shown on the earlier slide. On the right now, we independently assigned the toggle switch tryptophan for complexes with antagonists and agonists, and we see that the position or chemical shift of this toggle switch tryptophan is different, indicating local differences in the relative orientation of this toggle switch tryptophan to that PIF activation microswitch. Also, now, if you remember on an earlier slide, we were looking at some of the signals from the tryptophan residues on the intracellular surface and saw line broadening and signal multiplicity there. So if we look at those same tryptophan signals and compare the data for the native and the variant proteins, we see some interesting differences. So this slide shows a comparison of that data. So on the left, you have the native protein in complex with an antagonist, that's the blue spectrum, and the agonist, that's the black spectrum below it. And now comparing to complexes for the same ligands with the variant protein D52N. And these boxes that you see here, these dashed boxes with the data now are regions where we took projections of these signals. These projections are shown on the far right of the data, in which you see the signal multiplicity and broad signal for tryptophan 29 for the native protein, whereas we see a large increase in signal intensity for the variant protein D52N. And you can contrast that for the signal that we observe for tryptophan 143, which is essentially the same between the native protein and the variant protein. And now below that, we see a similar effect for the complex with the agonist, whereas we see a very broad signal for tryptophan 29 for the agonist complex with the native protein. We see a much larger signal and much more intense signal for the tryptophan 29 complex with the variant protein, indicating that we have altered dynamics at the intracellular surface. But the conformation near the drug binding pocket is preserved because we don't see differences for assigned signals on the extracellular surface. And now this data is also recapitulated where we see for glycine 218, which is located on the intracellular surface, we see large changes between the native protein and variant protein for the same ligand complexes. So again, the colors of the spectra are the same, and the boxes indicate projections around the signal. And you can see in the projection panels, we see large changes in the signal multiplicity and intensities between the native protein and the variant protein, indicating we also have change in conformational dynamics at that location on the intracellular surface of helix 6, which normally undergoes large conformational changes between active and inactive states. So in this slide here, we have a summary of the effects that we see for inactivation of this allosteric switch mapped onto the NMR probes distributed throughout the A to AAR. So at the intracellular surface, we essentially see a native-like response to ligand efficacy or no change. At tryptophan 246, which is located just above this D52 site, we have an altered relative conformation of the side chain of tryptophan 246 with respect to this conserved PIF activation motif. And then at the intracellular surface, we have abolished or altered conformational dynamics. So in this study, we connect changes in the local structural environment around the toggle switch tryptophan 246 with changes in the conformational dynamics at the intracellular surface. This is summarized in this cartoon where we show effects of allosteric switch mapped onto A to AAR. On the left, we have the native protein, which ligand binding and signaling is mediated through tryptophan 246, and we observe some strong coupling between aspartic acid 52 and this toggle switch tryptophan. It's coupled to conformational dynamics observed at the intracellular surface, as indicated by the small arrows between the two yellow balls on the intracellular surface. For the variant protein D52N, what we see is that this coupling is changed between the toggle switch tryptophan and asparagine 52. This leads to a loss of conformational dynamics at the intracellular surface, and that correlates directly with a loss of signaling activity for the variant protein. So in summary, we've established a platform for stable isotope labeling of human GPCRs, and we've used it to look at one key problem, but many more additional questions could be addressed with this technique. We've been able to map efficacy-linked conformational changes in the human A to AAR and provide a more comprehensive global view of those changes. And we've been able to relate signaling, changes in signaling to changes in conformational dynamics at the intracellular surface, which are linked to local structural rearrangements around tryptophan 246 with respect to this PIF activation microswitch. With that, I would like to thank my advisors, Professor Wüthrich at the Scripps Research Institute, Professor Ray Stevens at USC, as well as a number of my colleagues at both locations and collaborators, Ken Jacobson and Zhanggu Gao at the National Institutes of Health, as well as funding from the American Cancer Society for a postdoctoral fellowship. And with that, I'd be happy to take any of your questions.
Thank you, Dr. Eddy, for that informative presentation. We will now start the live Q&A portion of the webinar. So if you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click on the Send button. We'll answer as many of your questions as we have time for, so let's get started. Our first question is, will this new approach be able to tell us more about GPCR complexes with drugs that have not been crystallized?
Yes, that's a great question. So most GPCR drugs are very difficult to co-crystallize with GPCRs. So because we can get now site-specific structural information of a human GPCR, we're not limited in what we study to ligands of very high affinity, which are typically used for crystallization studies. In fact, we can look at ligands that have affinity as low as in the millimolar range. So we should be able to use this technique to get information about the conformation of the receptor in complex with ligands that haven't been co-crystallized.
The data with the A2A adenosine receptor are fantastic. How will you extend this technique to studying other human GPCRs in the future?
Well, we're optimistic that the PICA expression system can be used to express other GPCRs. There's some precedence for this in the literature where people have systematically screened the expression levels of different GPCRs in yeast or Pichia pastoris. In addition to that, other techniques are emerging from other research groups whereby they're able to incorporate stable isotope labeling into insect or mammalian cell expression systems as well. So that may also open up additional doors for future NMR studies of different human GPCRs.
We have time for one last question. You were able to identify and assign a subset of the signals from the NMR data. Can we gain further insight from the other as yet to be assigned signals?
Yes, so one of the powerful reasons this approach is exciting is that we can use the other signals, even if we don't yet have their identity, to confirm the foldedness or three-dimensional conformation of the receptor. In addition to that, we're pursuing additional techniques using more sophisticated spectroscopy, as well as including carbon-13 labeling, to be able to assign even more of the signals that you saw. That will give us a denser network of NMR probes that will allow us to better and better refine our view of signaling and signaling pathways in GPCRs.
Well, thank you again. I would like to once again thank Dr. Eddy for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May 22nd, 2018. You'll receive an email from LabRoots letting you know when the webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That is all for now. Thank you for joining us. We hope to see you again soon. Goodbye.